Evolutionary gene loss may help explain why only humans are prone to heart attacks. Researchers at University of California San Diego School of Medicine say the loss of a single gene 2 to 3 million years ago in our ancestors may have resulted in a heightened risk of cardiovascular disease in all humans as a species, while also setting up a further risk for red meat-eating humans. The findings are published July 22, 2019 in PNAS. Atherosclerosis, the clogging of arteries with fatty deposits, is the cause of 1/3 of deaths worldwide due to cardiovascular disease. There are many known risk factors, including blood cholesterol, physical inactivity, age, hypertension, obesity and smoking, but in roughly 15% of first-time cardiovascular disease events, CVD, due to atherosclerosis, none of these factors apply. A decade ago, Nissi Varki, MD, professor of pathology at UC San Diego School of Medicine, with co-author Ajit Varki, MD, distinguished professor of medicine and cellular and molecular medicine, and colleagues, noted that naturally occurring coronary heart attacks due to atherosclerosis are virtually non-existent in other mammals, including closely related chimpanzees in captivity, which share human-like risk factors such as high blood lipids, hypertension and physical inactivity. Instead, chimp heart attacks were due to an as-yet unexplained scarring of the heart muscle. In the new study, the Varkas and Philip Gortz, Ph.D., assistant professor of medicine, and others report that mice modified to be deficient, like humans, in a sialic acid sugar molecule called NEU5GC showed a significant increase in atherogenesis compared to control mice who retained the CMAH gene that produces NEU5GC. The researchers, members of the Glycobiology Research and Training Center and/or the Center for Academic Research and Training in Anthropogeny at UC San Diego, believe a mutation that inactivated the CMAH gene occurred a few million years ago in hominin ancestors, an event possibly linked to a malarial parasite that, in their findings, the research team said human-like elimination of CMAH and NEU5GC in mice caused an almost twofold increase in severity of atherosclerosis compared to unmodified mice. The increased risk appears to be driven by multiple factors, including hyperactive white cells and a tendency to diabetes in the human-like mice, said Ajit Varki. This may help explain why even vegetarian humans without any other obvious cardiovascular risk factors are still very prone to heart attacks and strokes, while other evolutionary relatives are not. But in consuming red meat, humans are also repeatedly exposed to NEU5GC, which researchers said prompts an immune response and chronic inflammation they call xenosulitis. In their tests, human-like mice modified to lack the CMAH gene were fed a NEU5GC-rich high-fat diet and subsequently suffered a further 2.4-fold increase in atherosclerosis, which could not be explained by changes in blood fats or sugars. The human evolutionary loss of CMAH likely contributes to a predisposition to atherosclerosis by both intrinsic and extrinsic dietary factors, wrote the authors, and future studies could consider using this more human-like model. In previous work, the Varkas and colleagues have shown that dietary NEU5GC also promotes inflammation and cancer progression in NEU5GC deficient mice, suggesting that the non-human sugar molecule, which is abundant in red meat, may at least partially explain the link between high consumption of red meat and certain cancers. Interestingly, the evolutionary loss of the CMAH gene appears to have produced other significant changes in human physiology, including reduced human fertility and enhanced ability to run long distances.